Welcome to solverfy.
For this question, we have three different molecules. We have a pre-mRNA strand, we have an mRNA strand, and we have a mutated version of the mRNA strand, meaning it has a difference in the gene structuring between the regular RNA strand and the mutant form. So when we look at this question and the answers, we're looking for exons and introns inside of this mRNA sequence. So exons are the parts or the genes that are going to be expressed when we make the protein, whereas the introns are going to basically be the fillers or random sequences in between that aren't going to be used inside of the protein. You can imagine these to be like ads inside of a magazine, for instance. So if we consider our exons and introns in these molecules, our pre-mRNA is going to include both those exons and the introns. However, the pre-mRNA strand is going to undergo processing inside of the nucleus of the cell to be converted into the finalized mRNA strand. And that's going to include the process of alternative splicing and the removal of those introns. So here, the mRNA strand is going to be exons only. So where we can look for the difference between our introns and exons is going to be the difference in genes expressed in these two different molecules. So you'll see that the second gene and the fourth gene have been removed. So those are going to most likely be the introns of this gene sequence, whereas the genes that remain, 1, 3, and 5, these are going to be those exons that are going to be translated to make the protein we want to. This mutant form they give us here is just as a distraction, and that third gene that was cut out is an abnormality, causing it to be a mutant. So if 3 were really an intron, we would not consider this last strand to be a mutant. But because 3 is required to make up this correct mRNA strand, its removal is considered a mutation instead. And of course, in these two molecules, if you are wondering, on the left-hand side, we have the 5' cap, and on the 3' side, we have that poly A tail, which is going to provide that stability for the molecule. Keeping those in mind, the best answer choice here is going to be A, that genes 2 and 4 are going to represent introns inside this molecule, whereas genes 1, 3, and 5 are going to represent exons. So here you should pick answer choice A.